Good morning, student. The last lecture we have already discussed about the uh, introduction of Golgi complex, their history, their occurrence, and today in this lecture we will discuss about the structures and the function of Golgi complex. So, let's come to the point. Structure of Golgi complex. Let's start with the cisterning. The cisternae of the Golgi apparatus are about 1 micrometer in diameter. The 1 micrometer in diameter. They are flattened plate-like or saucer-like closed component which are held in parallel bundles or stacks, one above other. Means means in each stack, cisternae are separated by a space of 20 to 30 nanometer, which may be contain rod-like elements or fibers. Each stack of cisternae forms a dictosome, which may contain 5 to 6 Golgi cisternae in animal cells or 20 or more cisternae in plant cells. So there are two phases of uh, Golgi. One phase is known as cis phase and another phase is known as trans phase. So cis phase, convex side of this stack is convex side of this stack forming cis phase, and concave side of the stack forming trans phase. So uh, the the cis phase is located near the nucleus, and the trans phase is located near the plasma membrane of the cell. So this type of polarization is known as cis-trans axis of Golgi apparatus. So I think you will understand about this cis and trans phase. This is very important. Cis phase means receiving phase of Golgi. This phase receives the protein, enzymes, and other molecules, and trans phase is releasing phase. After processing, their packaging, the protein molecules and other molecules get released from the trans phase. So these are the differences. Their positions are also vary. Cis phase are I've already told about this. Cis phase is located near the nucleus, and trans phase located near the plasma membrane. Now, the second component, that is tubules. Tubules is a small round tubules arise from the periphery of the cisternae. Some of these enlargement at their end from the vesicles. So tubules may be a complex array of associated vesicles in tubes surround the dictosome and radiate. Now the third organelles, that is vesicles. The vesicles are 60 nanometer in diameter, and there are three types. First one is a transitional vesicles, second one is a secretory vesicles, and third one is the clathrin coated vesicles. So first one, that is a transitional vesicle. Transitional vesicle are basically a small membrane bound vesicles which are formed as the leaves. Leaves means bubbles, which are formed as leaves, form the transitional endoplasmic reticulum to migrate and convert the cis phase of Golgi. Second one is the secretory vesicle. Secretory vesicles are varied size, means their size may be varied in different size, and they are the membrane limited vesicles which discharge from the margins of cisternae of Golgi. So, and third one is the clathrin coated vesicles. They are again aspherical and about 50 micrometer in diameter, and their surface is they have a very rough surface. They are found at the periphery of the organelles, usually at the ends of the single tubules, and are morphologically quite distinct from the secretory vesicles. So this vesicles are very much important because these are the clathrin coated, because the clathrin coated vesicles are play very important role in the intracellular traffic of membrane and secretory products. Means these vesicles, clathrin coated vesicles, are very important because of in the you can say in the secretion. In secretion, this clathrin coated vesicles is very important. Now, these are the structure of the Golgi complex. So just yes. Now you can see this is the structure of Golgi complex. This phase is known as cis phase, means receiving phase. Okay, and this phase is known as trans phase. As I have already told you, uh, this phase, this is a this is cis phase, and these are the small circular bubbles like structure are known as transport vesicle. These are the transport vesicle. These are and these are the cisternae. These are the saucer shape. These are the cisternae, and and these are the secretory vesicles. These are the just I have told you the clathrin coated. These are the coated vesicles. Means these are the clathrin coated vesicles. So these are the uh, this is the structure of Golgi complex. This is the receiving phase. The protein or any secretory product, hormones or whatever, they get received by this phase. In this Golgi complex, they receive received by this phase, this phase, and after processing, their compartment and their processing means certain packaging or synthesis or any or other after processing, this biomolecules can be released by this phase. This is a trans phase. Okay. So now these are the also structure of Golgi complex. We have a microscopic structure. Now we will discuss about the functions of Golgi complex. So in the next lecture we will discuss about the functions of Golgi complex. Thank you.